Carbon dioxide can also cause a change in the oxygen hemoglobin dissociation curve. Again, in red is the normal curve. Blue is showing a right shift, and the green is showing a left shift. If you look at the CO2 values, you'll see that the blue curve shows a partial pressure of CO2 at 61 millimeters of mercury. The normal millimeters of mercury, or partial pressure for CO2, is 35 to 45 millimeters of mercury. So the blue curve is showing you a partial pressure of CO2 that is significantly higher. When would the cells in a tissue bed be making more CO2? When they are having a higher metabolic rate. As they are more metabolically active, not only are they consuming more oxygen and generating more acids like hydrogen, like we saw in the previous dissociation curve, but they are also going to be making more CO2. CO2, like I said before, is another type of acid. Taking all of this together, there is going to be a right shift, and that means a more reduced affinity. The iron is more likely to be in a tensed or tented position and more likely to give off more oxygen. Again, it's a perfect setup. If the cells have more metabolic needs, more metabolic demands, then they are going to create a tissue bed with an environment that causes the hemoglobin to become more tensed, or the iron in a more tense position, and distribute the oxygen. The green shifted curve shows the CO2 is showing that the iron would be in a more relaxed position, more likely to capture oxygen and not let it go. The green shift shows a low pCO2. Taking the previous slide and this slide together, when a tissue bed is more metabolically active, it would be producing more acids, think hydrogen, and more CO2. If there are more hydrogen ions and more CO2, think right shift. Easier to unload, harder to load. The iron would be in a tensed position, wanting to give away the oxygen to the needy tissues. The green shifted curve shows a greedy hemoglobin with iron in a relaxed position, more likely to load the oxygen, less likely to give it up. What about temperature? Again, with a tissue that is more metabolically active, temperature is likely to rise. Heat, of course, is a byproduct of metabolic respiration. When you look at the normal red curve, you see a typical body temperature of 37 degrees centigrade. The right shifted blue curve shows a higher body temperature. A higher body temperature would be representing a tissue bed that is more metabolically active. The iron would be in a tensed position, more likely to let go of the oxygen, a lower affinity. The green curve is showing you the iron in a relaxed position, higher affinity, less likely to let go of oxygen, more likely to bind to it. Tissues that are not metabolically active do not release energy in the form of heat, not as much as tissue beds that are metabolically active. Again, right shifted, less affinity, more likely to let go. Left shifted, higher affinity, more greedy. What about the fourth condition, 2,3-DPG? 2,3-DPG is a byproduct of metabolism as well, specifically anaerobic metabolism in the red blood cell. Remember, the red blood cell lacks mitochondria, so it would rely on anaerobic respiration. Within a red blood cell, when 2,3-DPG levels go up, this would lead to a shift to the right, a right-shifted curve. A right-shifted curve, again, means a less greedy hemoglobin, a hemoglobin with less affinity for oxygen. This is an internal regulator for the red blood cell, an internal regulator that dictates how the hemoglobin will behave. Again, right-shifted, less greedy, more giving, more likely to deposit oxygen to the tissues. A left-shifted green curve, shown here, means a higher affinity. It means iron that would be in the relaxed state, more likely to bind. Right-shifted, tensed iron, more likely to give away. Left-shifted, green curve, relaxed iron, more likely to bind, higher affinity. In the top portion of this picture, I want you to see that the hemoglobin is shown here as alpha-2, beta-2, adult hemoglobin. And the DPG molecule, shown here as a black oval, binds only to the beta-globin chains. I need you to remember this, especially as I go to the next slide. The 2,3-DPG only binds to the histidine residues in the beta-globin chain. This is going to explain why we see different affinity curves, different shifts for adult hemoglobin versus fetal. If I were to actually take red blood cells from a fetus and red blood cells from an adult, lyse open the red blood cells and harvest only the hemoglobin, the fetal and adult hemoglobin would show exactly the same affinities. Yet in vivo, in a living red blood cell, we would see that the fetal hemoglobin is left-shifted. Left-shifted means higher affinity. Higher affinity for oxygen means the iron is in the relaxed state, wants to bind to oxygen. Fetal hemoglobin, you remember me saying, has a higher affinity than adult. The reason why it has a higher affinity than adult hemoglobin in vivo is because fetal hemoglobin has the globin chains of alpha-2, gamma-2. Adult has alpha-2, beta-2. And on the previous slide, I showed you that 2,3-DPG binds with the beta-globin chain. Fetal hemoglobin does not have the beta-globin chain. That means when 2,3-DPG levels are elevated, only adult hemoglobin will be affected. Only adult hemoglobin has the beta-globin chain. This means for a woman carrying a fetus, maternal hemoglobin means adult hemoglobin. It means that she has the beta-globin chain. The iron would be in the tensed position, less affinity for oxygen, more likely to give off oxygen to the tissues, less likely to load. Fetal hemoglobin is the reverse. It would have iron in the relaxed position, more likely to load with oxygen, less likely to give off to the tissues. Notice that I'm saying less likely, more likely. I am not saying that fetal hemoglobin does not give off oxygen to the tissues. Clearly, that's not the case. Otherwise, none of us would be here. None of us would be listening to this lecture or giving this lecture. Less likely. Fetal hemoglobin requires a greater change in the partial pressures for oxygen to release oxygen compared to adult hemoglobin / maternal hemoglobin. I demonstrated this on an earlier slide. So I've talked about how red blood cells, or what I should say, red blood cells need for proper synthesis and maturation. Included in proper synthesis of red blood cells was a very lengthy discussion on the ingredients needed for proper hemoglobin. What would we expect for proper red blood cell removal? We forget that red blood cells are technically not true cells. They don't have a nucleus. They don't have organelles, for crying out loud. And as such, they have a lifespan of approximately 120 days because they don't have a nucleus. They don't have the ability to have new gene expression to repair or renew proteins that might have been damaged. They don't have organelles like mitochondria. They cannot participate in oxidative phosphorylation. So red blood cells, erythrocytes, they only live for about 4 months, 120 days. They sustain incredible mechanical stress from passing through narrow capillaries where they have to pretty much fold in half to get through the diameter, the lumen of the capillary. They have limited ATP stores because they cannot participate in aerobic respiration. They can only participate in anaerobic respiration, so the levels of their ATP will be compromised. Because they carry oxygen, they are subjected to a significant amount of oxidative damage. They don't have organelles to generate new proteins like the nucleus, the endoplasmic reticulum, ribosomes, to replace those proteins that can in turn manage oxidative damage. So their membranes become more frail as there's more oxidative damage on the lipids found in the membrane. Proteins also sustain oxidative damage. The cells become progressively more fragile. Where are red blood cells removed? They are removed in both the liver and the spleen. In the liver, hepatocytes will help with the removal of the red blood cells. More on this in a moment. The actual cells that phagocytize, engulf the aging red blood cells are called macrophages. They are found in both the liver and the spleen. In this picture, I am showing you a macrophage. I'm circling it now. This macrophage has 4 red blood cells engulfed. 1, 2, 3, 4. Again, macrophages are found in both the liver and the spleen. Macrophages are not the same as hepatocytes, a cell I just mentioned that is found in the liver. Hepatocytes are the functional unit, if you will, in the liver, the functional cell. Macrophages happen to be a secondary cell found in the liver. Between the macrophages and the hepatocytes, we will see red blood cell removal, destruction, and some parts of the red blood cell recycled. Let's continue on that in the next slide. Shown here in blue is a senescent red blood cell. Think of that as an aged red blood cell. Shown here in yellow, this entire circle shows a macrophage. The macrophage phagocytizes, engulfs the aged red blood cell. When it engulfs the aged red blood cells, it will break through the phospholipid bilayer and it will release the globin chains. The globin chains can be released into the plasma as free amino acids. Free amino acids can be used by any cell in the body for synthesis of new proteins, for example. Also, when the red blood cells are destroyed, we get release of the heme groups. And I'll remind you that heme is the porphyrin ring and the iron. Well, the iron part can be released from the macrophage to a plasma protein called apotransferrin that I talked about before. And when iron binds to apotransferrin, that protein, its name, now turns into transferrin. Transferrin can then deliver iron to cells in the body, and they can store that iron bound to a cytoplasmic protein that is called apoferritin until iron is bound to it, and then it turns into ferritin. Ferritin is the healthy way to store iron in our cells. The abnormal or unhealthy way to store iron in our cells in a crystalline form is called hemosiderin. That means if I think of the globin chains as being recycled to amino acids, and the iron from the heme group being recycled to cells of the body and stored as ferritin, only the porphyrin ring, which is the part of the heme group that cannot be recycled. The macrophage will exocytose the porphyrin ring, and when that porphyrin ring exits the macrophage into the plasma, it is released as free bilirubin. Free bilirubin, let me write this up here, is not very water soluble. Free bilirubin will bind to albumin, a plasma protein, our most abundant plasma protein. Albumin will envelop the free bilirubin and make it more water soluble. Albumin in this capacity is acting like a carrier protein. Albumin will carry the free bilirubin to the hepatocyte. Now this is a cell within the liver. Remember, macrophages are found in the liver. They're also found in the spleen. They're found in many tissues in the body, for crying out loud. Albumin might only be transporting this free bilirubin a short micrometer away to an adjacent cell that happens to be a hepatocyte if this macrophage was found in the liver. Regardless, when the macrophage releases the free bilirubin and it binds to albumin, the albumin will now relinquish this free bilirubin to the hepatocyte in the liver. Within the liver, within the hepatocyte cell, glucouronic acid, shown here as this purple circle, is then bound to the free bilirubin. The two are bound together. We say they are conjugated. And when the two are bound together, we say we have created conjugated bilirubin. The hepatocyte can then release the conjugated bilirubin as a byproduct into the bile secretions. Remember, hepatocytes make bile. Bile is stored and concentrated in the gallbladder. Bile is excreted out of your digestive system by going from the bile duct, the common bile duct, into your small intestine. While it's in the small intestine, bacteria there can convert the bile into other forms called stercobilinogens, which is the reason why your feces look brown. Some of the conjugated bilirubin, because it is more water soluble, so conjugated bilirubin is more water soluble. Some of that conjugated bilirubin, while it's waiting in the gallbladder being stored and concentrated, some of it can be reabsorbed into the plasma. And while it's in the plasma, your kidneys can filter that conjugated bilirubin. And while that conjugated bilirubin is going through your renal system, it's converted into urobilinogens. And while it's going through your renal system, there are bacteria there that will basically process this conjugated bilirubin into urobilinogens, and the urobilinogens will take on a color. This is why your urobilinogens are yellow. So urobilinogens make your urine yellow. Stercobilinogens make your feces brown. You see, it is dying aged red blood cells that make your feces brown and your urine yellow. That's important to know, especially when we're studying about jaundice. Jaundice means that you have a higher amount than normal of bilirubin. When we actually draw blood and collect urine from a patient, we measure the levels of bilirubin from both of these body fluids. We actually distinguish between conjugated and unconjugated bilirubin. I'll remind you, unconjugated bilirubin is released from the hepatocyte. It represents the porphyrin rings that are not more water soluble. Conjugated bilirubin represents porphyrin rings that have been bound to glucouronic acid. This only happens after the unconjugated bilirubin enters the hepatocyte cell. Only if the hepatocyte cell has normal function can the glucouronic acid be joined, conjugated, bound to the unconjugated porphyrin ring. Different people can show with jaundice, present with jaundice. When someone has jaundice, their skin looks very yellow. And the yellowing of the skin is because of the bilirubin accumulation. The yellowing of the skin can be because of either free bilirubin or conjugated. What are the different populations that could have jaundice? A common population for jaundice would be in newborn babies. We say this is called jaundice of the newborn. Jaundice of the newborn is a problem with hemolytic jaundice. The baby that is born has an immature liver. The hepatocytes are immature. They do not have the capacity, not at least very well, to conjugate the glucouronic acid to the porphyrin rings. Remember, porphyrin rings equals free bilirubin. So with hemolytic jaundice, we now have a newborn that is outside of the mother, is not exposed to all of the good pregnancy hormones that propagate more red blood cell synthesis and ensure a longer lifespan. We also have new red blood cells being synthesized because the hemoglobin is converting from fetal hemoglobin to adult. So there's turnover of the red blood cells. This turnover is happening more quickly than what the hepatocytes can process. So in hemolytic disease of the newborn, in hemolytic jaundice of the newborn, I should say, we see a higher level of unconjugated bilirubin. Again, it's because the liver cannot keep up with the turnover of the red blood cells. This is different than what we see in obstructive jaundice. Obstructive jaundice happens because we have clogged bile ducts, maybe because the person has portal hypertension. I'll remind you that we do see portal hypertension in patients with kwashiorkor, where their albumin levels go down. Albumin is made in the liver. If the liver is compromised in its function, we will not be able to see the unconjugated bilirubin being conjugated to the conjugated bilirubin. And also, if the liver is not making albumin, we can see this ascites accumulating in the peritoneal cavity. We learned about this in our body fluid compartments lecture. So in obstructive jaundice from portal hypertension, we can see that the bilirubin might be the most abundant of the conjugated type, meaning that they're, well, let me back up. If it's truly obstructive and it's from, let's say, a gallbladder stone, a gallstone, then there's nothing wrong with the liver, is there? So if it's truly obstructive jaundice from a gallstone, and we assume normal, healthy hepatocyte function, then there is no problem with the hepatocytes converting unconjugated bilirubin into conjugated. The problem is getting the conjugated bilirubin excreted out of the body with the bile. In these patients, if we were to draw their blood, we would see that their conjugated bilirubin is higher than normal, reflecting it's a problem getting it out of the body. However, if it is a patient, say an older patient, with a dysfunctional liver, maybe they have cirrhosis of the liver, maybe from drinking too much, we would expect to find jaundice in that individual. In that individual, we would expect to see higher levels of unconjugated bilirubin and conjugated bilirubin. We would expect to see, with the destruction of the hepatocytes or failure of them, the unconjugated bilirubin going up, but also as the liver is failing, levels of albumin would go down, and their edema in their peritoneal cavity would go up, and this would obstruct bile ducts. So in a person with liver failure, expect them to have jaundice for both kinds of reasons. By that I mean elevated unconjugated bilirubin as well as elevated conjugated bilirubin. When we have a bruise, an echimosis, we think of the wonderful colors that are produced by this bruise. We see yellows, we see greens, we see some purples. All of those colors reflect damaged red blood cells within the tissue bed undergoing this removal of the bilirubin, removal of the porphyrin ring. So that green represents bili verde, yellow represents the bilirubin, and as we see that green transitioning to yellow and the yellow fading away, it represents macrophages removing those damaged red blood cells from that region, removing the unconjugated bilirubin from those red blood cells, putting it into the plasma, and then ultimately the hepatocytes removing that unconjugated bilirubin from the plasma. In our next segment of videos, I will discuss, at length, our red blood cell disorders. I will be talking about polycythemia and anemias.